We can wait for a few more minutes while we let the other participants in. Okay, we will start in 2 minutes. We're just waiting for more participants. Okay, we can start now. Thank you. Welcome everyone from wherever you are. Good morning, good afternoon, or good evening. Thank you all for joining us today for today's webinar, Gene Drive Technologies, Advances in Health, Conservation, and Governance, organized by the International Service for the Acquisition of Agri-Biotech Applications in partnership with the Outreach Network for Gene Drive Research. We're based in the Philippines. Actually, ISA is based in the Philippines, and we are working towards advancing biotechnology through knowledge sharing, capacity building, and policy support to farmers and consumers all over the world. And we're a nonprofit charitable organization based in the Philippines. Okay, this panel aims to contribute to informed, balanced, and inclusive discussion by highlighting latest developments in gene drive science, governance, and stakeholder engagement. This webinar will revisit core concepts while offering timely updates on real-world progress, emerging policy discussions, and evolving societal considerations shaping the gene drive landscape. I am Dr. Rodora Romero Aldemita, the Executive Director of the AISA based in the Philippines, the AISA Southeast Asia Center, and the Director of the Global Knowledge Center on Crop Biotechnology. And I'll be moderating the whole webinar. So now, before we start, I would encourage all of you to display your full name and affiliation or on your Zoom profile if you are able. And to do so, please click the three dots on the right-hand corner of your video box. There you will find the option Rename. Okay, we'd like to see that. And currently, we are now 90 participants, and we hope to reach about more than 100 since we have more than 200 registrants. And so for a little bit of housekeeping, can you share now our intro slide? This webinar is recorded, and the recording may be shared online at a later date. We're also live on Zoom, on Facebook and YouTube. And so please put your questions in writing in the Q&A box and not in the chat, okay? It's easier to see it in the Q&A box, and you can only put important reminders in the chat. There is time for answering them after the presentations, but speakers are also encouraged to reply in writing during the webinar so that we can answer as many as possible. You can direct your questions to a specific speaker or keep it general, and apologies in advance if we are not able to answer all questions. Now let's go to the objectives. So the activity objectives, of course, is to provide an overview of gene drive technology and its potential applications, and then provide an update of the state of gene drive research and development, highlight the evolving regulatory landscape, and lastly, explore emerging approaches to stakeholder engagement. With that, we can start with the first speaker. I think, is that the first speaker. Let's get started with our first presentation by Dr. Jackson Champer. He is the principal investigator of Peking University, and Dr. Champer holds advanced degrees in physics and biology and leads research on gene drives and evolutionary genetics. Let's give a round of applause to Dr. Jackson Champer, and you're on now.
Hello, everybody. I'd like to tell you a little bit about gene drive. So first of all, what is a gene drive? There are actually both natural and engineered versions of gene drive that in many ways are very similar to each other, but today we're going to focus mostly on engineered gene drives. So what's the concept behind a gene drive? We make a few gene drive individuals in the laboratory, and then we release them here in red into a wild population here in blue. Over time, the gene drive will spread through the whole wild population until, ideally, everyone has it. So how does that work? Normally, with Mendelian inheritance, this single allele here in orange is inherited by half of the offspring. So that means if there's no fitness costs, we expect that allele to stay at the same frequency in the population over time. A gene drive allele, though, is special. Even though this gene drive is in a heterozygote, so this organism has only one copy here in red, you notice that all of the offspring inherit the gene drive. This means that over time, the gene drive will increase in frequency in the population. With just a small release in just a few generations, the gene drive can reach the entire population. So this is a potentially very powerful strategy for using genetics to alter an entire wild population. So what can we do with this? Well, there's quite a few different applications. One of them is population modification. We insert a gene together with the gene drive, a cargo gene, and then when we spread the gene drive through the population, we'll also spread this desired cargo gene. We can also configure a gene drive to suppress or even eliminate the target population. We'll talk more about that later. One of the most interesting applications of gene drive, of course, is in mosquitoes. We already have cargo genes for many nasty vectors, such as Anopheles mosquitoes that spread malaria and Aedes mosquitoes that spread a variety of viral-based diseases. And then there's also Culex mosquitoes, which are bad, but not quite as bad as these two. But there's a lot more we can do with gene drive besides trying to fight against these vector-borne diseases. All over the world, there are various invasive species that are causing havoc in many ecosystems, especially those in islands. With a gene drive, we could potentially suppress these species where they're causing problems. And of course, we've got agricultural pests represented here by the spotted-winged Drosophila. With a gene drive, we could suppress these creatures in agricultural lands, thus having a much more environmentally friendly method for pest control. So how do these gene drives actually work? Well, it's important to realize that there are actually multiple types of gene drives. The homing drive, which I'm showing you here, is just one of them, but it's perhaps the most powerful. It uses CRISPR-Cas9 to cut a wild-type allele, a normal chromosome, opposite the gene drive allele. Anytime DNA is cut, it's going to need to be repaired. But cells are very good at that. In this case, the cell finds DNA on either side of the cut and matches it to identical DNA on either side of the gene drive allele. This means that during homology-directed repair, the gene drive allele will be copied into that other chromosome. So this happens in germline reproductive cells. So even though most cells in the organism are heterozygotes with just one copy of the gene drive, the germline cells, which are the ones passed on to the offspring, are homozygotes with two copies of the gene drive, and thus all the offspring will inherit the drive. Unfortunately, this homology-directed repair is only one potential mechanism. If we get repair by end-joining, that can change the target sequence, creating a resistance allele. This resistance allele sequence is different from the guide RNA sequence of the Cas9, so it can no longer be cleaved and no longer converted to a gene drive. These resistance alleles are one of the biggest technical challenges in making a gene drive that is actually good enough to work in the real world. And here's why. If we have a modification gene drive, even if the gene drive gets to 95% or more of the population, if that resistance allele has a fitness advantage, over time, it will outcompete the gene drive. For a suppression gene drive, it's even more dramatic. Here, that resistance allele will increase in frequency almost as fast as the gene drive did, and that means the gene drive will immediately fail. So, how does suppression work in the first place? This is a special type of gene drive that targets an essential but haplosufficient female fertility gene. So how does that work? In somatic cells, we have one gene drive allele and one wild-type allele. So these females are fertile, even though in their germline, they're converting the wild-type allele to a gene drive allele. So they can still pass on that gene drive allele to a lot of offspring. So eventually, the gene drive allele increases in frequency in the population until almost everyone has a gene drive. And then we'll have a lot of female gene drive homozygotes. Those homozygotes will be sterile females. That means they can't successfully reproduce. And that's why when the gene drive increases in frequency to a high level, it will suppress the population. In our first try, we made sure that we would avoid the worst kind of resistance alleles by using multiple guide RNAs to target our female fertility gene. In this case, we did it in Anopheles stevensii, which is a very invasive urban vector of malaria. But as you can see for drive inheritance, instead of getting near 100% of the offspring with the drive, we only got about 3/4 of the offspring with the drive. So that's not enough for a strong drive. We had to upgrade our gene drive. And with this new gene drive, almost all of the offspring can inherit the drive. So we think that this gene drive will give us enough power for population suppression while still being able to avoid resistance with multiple guide RNAs. So we're testing this gene drive in more detail now. Gene drives have also been made in a variety of agricultural pests. We're doing collaborations with some of these, though we haven't published in it yet. One example is the spotted wing Drosophila, which uses a serrated ovipositor in females to cleave into fresh fruit, laying their larvae. There's also the medfly, which had a very interesting homing drive, and the diamondback moth, where gene drives still need to be developed to have good efficiency. And scientists are also investigating some other agricultural pests besides these, but only these three have actually had peer-reviewed publications so far. Of course, one big problem with gene drive is the need for confinement. That homing drive I talked to you about is so powerful that if a gene drive spreads in one spot of the world, pretty soon it's going to be everywhere that that species exists, and that can cause problems. We don't want to suppress invasive species or agricultural pests in their native habitat, for example. So to do that, we can make a confined gene drive. I won't go into it in too much detail, but basically for a confined gene drive, if we only have a little bit of the gene drive, it's just going to go away. But if we release enough of the gene drive, it can spread in the target population, but migrants to a distant population will never get to a high enough level to spread the gene drive because it's constantly being removed. We made a gene drive like this called a tear drive, also in mosquitoes. We've measured it for based on mathematical modeling, we at a high enough here in pink, the drive will spread. If it's released at a low level, the gene drive will naturally be eliminated. We also have self-limiting gene drives. So these are basically temporary gene drives that will go away on their own. You can release them a lot, like in sterile insect technique, and that will start suppressing the population and increasing the frequency of a gene drive. But as soon as you stop releasing over here, well, then the frequency of the gene drive will slowly decline and the population will recover. The advantage of this is that compared to sterile insect technique, which has huge release sizes, 4.3 even in this perfect model, or Riddle, which is a little better, but still requires high release, if we have a gene drive, we can get the release at least 10 times lower and still eliminate the target population. All right, that's all I wanted to tell you about today. Many thanks to my lab members who are responsible for a lot of the data that you saw here. Feel free to ask me questions later in the presentation.
Thank you very much, Dr. Champer. Actually, this is our basic information on gene drive, and I know that you have our audience, which is composed not only of academics, but also of the general public would like to know more. So we will take note of your questions later. Please put your questions in the Q&A box. And thank you so much again, Dr. Champer. Thank you. Let's give him a round of applause, the virtual one. So now let's go to our next speaker, who is Dr. Brian Tarimo, to share about the recent advances in gene drive for vector-borne disease control. Dr. Tarimo is the lead scientist and Transmission Zero in Tanzania. Dr. Tarimo specializes in molecular biology and bioinformatics applied to malaria research, with expertise in transmission biology, transgenic mosquitoes, and biosafety. Let's have you on board, Dr. Tarimo.
Thank you, Rodora. Let me share my slides. Yes, good morning, good afternoon, good evening, everyone, from wherever you are. Briefly introduce, I'm Brian Tarimo from the Ifakara Health Institute and I'll briefly talk about advances in gene drive for vector-borne diseases control. So why focus on vector-borne diseases? 80% of the world's population is at risk of one or more vector-borne diseases. 17% of the global burden in communicable diseases is due to vector-borne diseases, and there's over 700,000 deaths caused by vector-borne diseases annually. Now, the response frameworks has several pillars which range from strengthening inter and intersectoral engagement, surveillance, monitoring, and scaling up of existing interventions. But one of the foundations of each and every of this pillar is the increase in basic and applied research and innovation. And today we'll look at briefly with regards to this one innovation, which is the gene drive for the control of these vector-borne diseases. So briefly, before I go into what gene drive is and how this is being applied in control of vector-borne diseases, I'll briefly talk about mosquito genetic transformation. And mostly what I'll talk about has been briefly highlighted by the previous speaker. Why genetically modify mosquitoes? The current tools that we have to control vector-borne diseases, bed nets, which contain insecticides, they've reached their limit of efficacy. There's resistance being developed in bed nets and in spraying. Habitat management and removal is quite cumbersome. Spraying is quite fumigation is quite cumbersome, and therefore there is a need of looking of some of these new innovative tools. Sorry, try to move. Now, sorry, I'm unable to move forward. I don't know why.
Okay, EJ, can we put up his slide? Do we have his slide?
I have slides. I'm trying to.
No, I'm talking to EJ, our technical person. Maybe we can stop share and we'll share your slide from our end.
Let me try again.
Please.
Okay, so I do. Okay, let me stop sharing.
Okay.
Sorry about that.
Can you share it again from your end? And then if not, then we'll do ours.
There,
Oh.
you can see.
Yes, and it's not moving still?
It's not.
Okay. EJ, can you share his slide? You can stop sharing now, Jax. Hi, Brian. There. Sorry about that. So while we're waiting, we'd like to invite everybody to put your questions in the Q&A box, the Q&A function, and we'll get to them later. And so the first presentation was on the presentation of Dr. Jackson Champer on gene drive technologies and potential applications. We're now in the presentation of Dr. Brian Tarimo on advances in gene drive for vector-borne disease control.
Okay, I can do that. I think it worked again. Can I continue?
Oh, okay. Okay, wonderful. Thank you. Yes. Can you?
Okay, so I was saying that, as I was saying that the current tools that we have really reached their limit of efficacy. The insecticides that are being used in bed nets and sprayings have developed mosquitoes have developed resistance against this. Fumigation and habitat removal tends to be cumbersome and brings pollution. Screens have holes, and therefore there is a need to look at some of these innovative tools to control these mosquito vectors. Now, if you look at what makes a mosquito a good vector for malaria, any other vector-borne diseases, if we look at factors such as host feeding, vector susceptibility, that the immunity to parasite, the longevity, that they're able to live long enough to lay eggs, and these eggs can mature to the next generation. The density, they produce high rate of eggs, and the habitat preference, they live close to human beings, therefore they have access to a vertebrate human blood feeding. If we look at all of these factors, we tend to realize that they do have some genetic component. Now, this means that there is somewhere along this the vector genome, the mosquito genome, that there's a gene that is makes it live long enough, or control its feeding behavior, or controls the parasite development, innate immunity, and also reproduction. Now, if we couple this with some of the genetic tools that we've had over the years, we know the genome of these mosquitoes. Anopheles gambiae genome came out in 2002, and others followed thereafter. And if we look at this technology such as RNA interference, and most recently the CRISPR-Cas9 technology for gene editing, we're equipped with the tools to be able to genetic modify these genes, and therefore come up with mosquitoes that are genetically modified and to control vector-borne diseases. However, so we know we can modify genes such as resistance to parasite, insecticide susceptibility, change behavior, or sterility, but we're still faced with the problem. If you look at the slide that's up on the board, the yellow mosquito is the one that's genetically modified. As we go from generation 1 to generation 2 to later generations, we find that the number of these modified mosquitoes compared to wild-type mosquitoes is not increasing. Now, if we want to have any effect of controlling vector-borne diseases, we want a substantial number of these genetically modified mosquitoes to be in the general population. As we can see that the frequency remains low and it's not increasing. So this is a problem that has to be solved. And this is because that there's Mendelian inheritance, as Jackson earlier said, that there's a 50-50 chance of this gene being inherited between the parent lines. And therefore, as a result, our altered gene is not spreading from one generation to the next. Now, if we want to solve this problem, and if we have these genetically modified mosquitoes for control of vector-borne diseases, we need to think of another strategy, or another way of ensuring that a significant number of these mosquitoes in the wild population contain the altered gene. And with this is where gene drive technology comes in. Gene drive technology, basically, it's a natural genetic phenomenon. It is any genetic element that is able to bear its own inheritance, and it requires sexual reproduction. As we have on the slide here, you can see that on the left, the normal inheritance, which is the Mendelian inheritance, there's a 50-50 chance of a genetic element being passed from the parental line to the offspring line. While vis-a-vis the gene drive, there's a nearly 100% chance of a genetic element to be passed from the parental line to the offsprings. Now, this is ideal if we have altered gene in our mosquitoes. This will enable us to ensure that our altered gene is able to spread from generation 1 to generation 2 to generation X, and therefore a significant number of these mosquitoes will have this altered gene, and therefore bring a significant chance of controlling the disease, and therefore bring down transmission of this disease. As you can see in the slide, the yellow or orange circle, that's our altered organisms. And from generation 1 to the later generation, their number significantly increases compared to the rest of the population. And therefore, the frequency of the altered is increased, and therefore bringing about the control of the disease that we want. Here's another slide that shows that. And you can see on the left is Mendelian inheritance, which is the altered gene is not spreading. On the right is the super Mendelian inheritance or the gene drive inheritance in mosquitoes. And you can see that the alter gene is increasing from one generation to another, and therefore bringing about this effect that we want. And earlier speaker Jackson explained how this gene drive is being inherited from parental line to offsprings. Now, for control of vector-borne diseases, we have two options. We have two options or two strategies. One on the left is population suppression, whereby you introduce wild-type mosquitoes and therefore bringing down or suppressing the population of mosquitoes. This should not be confused by eliminating. You suppress the population of mosquitoes to such a number that it is low enough that it blocks transmission of these vector-borne diseases. And this other strategy is population modification. Others tend to call it population replacement, whereby you introduce alter genes into the mosquito population, and therefore these mosquitoes are unable to transmit the pathogen that they carry. For example, with malaria, they're unable to transmit Plasmodium, and therefore bringing down transmission of malaria. You can see here, population suppression, you disrupt an essential genes, for example, is susceptibility to insecticide or reproduction, therefore making these mosquitoes sterile, and therefore you reduce their reproductive capacity. And on the right, with the population modification, you introduce a gene, you integrate this gene into the mosquito genome, for example, the anti-malaria effect, and therefore these mosquitoes are unable to transmit the disease. So if we look for gene drives for control of vector-borne diseases, and particularly malaria, if you look, Africa bears a substantial burden of this disease. And who works on gene drive mosquitoes in Africa? There are several players. As earlier mentioned in my introduction, I'm from Tanzania, and in Tanzania, through the Ifakara Health Institute, we work with Transmission Zero project, which focuses on mosquito replacement. And in the later part of my presentation, I'll briefly talk about the project. So about the Transmission Zero project, who is Transmission Zero project? It's a collaboration between the Imperial College London, Ifakara Health Institute in Tanzania, National Institute of Medical Research here also in Tanzania, and the Swiss Tropical Public Health Institute. We focus on population modification. Our transmission work is carried out at Haichai, the site called Bagamoyo, which is an eastern part of the country. And also our potential field work is an island called Ukerewe, which is located within Lake Victoria. As you can see on the map on the left here, Lake Victoria is located within the 3 East African countries. And on the Tanzanian side of the lake, there's Lake Ukerewe, where this is the potential field site for release of the genetically modified mosquitoes that we're working on. So what did the project do? So we have introduced what we call MMCP strain. These are expressing antimicrobial peptides, 2 antimicrobial peptides, maganin and melittin, which have been introduced into a carboxypeptidase gene within the Anopheles gambiae genome. So earlier studies have shown maganin, which is from African frog-lock. It's a peptide from African frog-lock, and melittin, which is from the European honeybee. These 2 toxins, when they were introduced along with the blood meal, infected blood meal, and fed to mosquitoes, they were shown they have significantly reduced the population of oocysts. So what we have done or managed to do is to introduce these 2 genes into the zinc carboxypeptidase gene, which is in the Anopheles genome. And therefore, these 2 genes are being expressed. And therefore, once they're expressed during after blood feeding, they're able to reduce oocyst population in the mosquito and therefore block transmission. I would like to highlight what we've introduced is what we call a very tiny genetic modification. If you look at the Anopheles genome, it's about 270, 280 megabase pairs. We've only introduced about 0.8 kb, so it's a very, very tiny modification within the genome. So this is a brief or what the mosquitoes do, or what so the meganin and melittin through the central dogma, they're being expressed, and once they encounter these oocysts in with or kinids within the mosquito midgut, they're able to block their development, and therefore this mosquito is unable to the parasites that they don't fully develop, and therefore this mosquito is unable to transmit the parasite. And some of the works that have been published, we've shown that the mosquitoes that have the MMCP at day 7, they have the oocysts are stunted in diameter, and they delay their development, and therefore when these mosquitoes go for a subsequent blood meal, that they are, and the oocysts have not ruptured, or they do not have sporozoites in the salivary glands, and therefore they're unable to transmit the disease. However, if for any gene drive mosquito for control vector-borne disease, they have to be a phased approach for testing and deployment. Our colleague John Connolly published in 2024, and also there's a WHO guideline of how this should be implemented. Currently, as a project, as Transmission Zero, I'd like to highlight that we're in contained phase. We have not gone beyond, or at the stage where we can go beyond the laboratory. All the work we've been done are in phase 1, which is in the laboratory studies. But after laboratory studies, there's phase 2, where you go for small-scale isolated release, and then there's phase 3, where you can do small-scale open release and large-scale, and then after there's phase 4, where you have to do post-implementation surveillance to ensure that what you've released into the environment, does it have any significant entomological and epidemiological effect? And there's different endpoints that you look at each phase to ensure that what you're working on, does it meet the criteria that of your evaluation. So this is how Ukerewe is, and this is the strategy that Transmission Zero is planning to work on. As I mentioned earlier, we're currently at the laboratory stage studies in contained facilities, but later on, we plan when we go for small-scale isolated release, we plan to release these mosquitoes with the effector strain only, meaning that they do not have the drive. Therefore, they will only be in an isolated area. They cannot spread. And then in the small-scale open release is released these 2 types of mosquito, one with the Cas9 drive and one with the effector. So once only those mosquitoes that have both the genes that will be able to drive, and therefore the scale or the spread will be limited, and therefore large-scale open release is to have with the effector with the release, and therefore to see spread of these mosquitoes across the whole island. So this is the approach that the project is currently working on. And with that, I'd like to say thank you, and I welcome your questions, and my apologies for the IT glitch earlier.
Thank you so much, Dr. Brian Tarimo. And actually, this part of the talk is the one which we know is going to give good impact, especially for vector-borne disease, and in Asia and in Africa now, and hopefully we'll have it also in Asia. Thank you so much. We'll have questions later, and please put your questions now. I see some questions, very interesting questions. Please bring them up and write them at the Q&A box. Now we're going to the next presentation, and we're happy to note that we're keeping our time. So I'd like to invite Dr. Gelshan Godahewa to take the floor and present about progress on gene drive research for invasive species and biodiversity protection. Dr. Godahewa is a postdoctoral researcher at University of Adelaide's Genome Engineering Lab, and he focuses on CRISPR-Cas9 applications for pest control and innovative mammalian gene drive strategies. Let's have you on board, Dr. Godahewa, or how do we call you? Dr. Gelshan.
Cool.
Thank you.
Yeah, let me share my slides. So now you can see the slides, right?
Yes, thank you.
All right. Thank you for the introduction, Ola. From the Jackson's talk and the Brian's talk, now we know what is gene drive and why we need to develop and how we develop the gene drive technologies. Today, I'll be talking about how we develop gene drive technology to control invasive mammal species. In 2023, there's a report came out. It is a very comprehensive report, and it consists on 86 experts are involved from 49 different countries to develop this report. It shows that invasive species are costing the world about 423 billion US dollars every year. Australia became one of the most affected countries in the world. Why? Because invasive mammals like feral cats, foxes, rabbits, rats, mice, and feral pigs are the biggest threat to the biodiversity loss in Australia. They have already contributed to more than 34 native mammal extinctions in Australia during the last few decades. As we know, invasive species don't just threaten the wildlife. They also create serious public health and also create major agricultural losses and damage the ecosystem. The bird shown here is the endangered grey-headed albatross living in Marion Island on South Africa. This species is heavily affected by the invasive predators like mice. As you can see here, this mice is eating the live bird. This figure, it shows the 100 of mice trapped during the mouse plagues in Queensland in Australia. And these species, like mice, can multiply quickly and cause severe economic losses and environmental damages. So the gene drive offers a species-specific and humane way to control invasive population without widespread poisoning or culling. Let's see how we develop the gene drives in mice. We are using the CRISPR-Cas system to generate transgenic animals. This system contains two main components. The guide RNA, which direct the Cas9 into the specific DNA sequence, and the Cas9 is an enzyme which acts like a molecular scissors to cut the DNA. So this system creates the double-strand break of the genome, and we can insert the gene drive component into that particular region. That's how we generate the transgenic individual. We have worked on several gene drive strategies to control invasive mice. The first one is the homing gene drive. Jackson explained how the homing gene drive works very well. So I'm not going to go deeper. So this system can pass the homing gene drive component more than 50% to next generation. This is extremely well working in mosquitoes, but much less efficient in mice. The second strategy we tried in the X-shredded gene drive, the gene drive component which shed the X chromosome during the sperm development. So the only Y-bearing sperms are alive. So the next generation will be mostly male population. However, in our X-shredded males, we observe the blocking of the sperm development rather generating the male bias population. In both cases, timing and level of the Cas9 is important. Our third approach uses the T haplotype and targets the female fertility to control the population. The T haplotype is a naturally occurring meiotic gene drive system in male mice. It consists main two versions of chromosome 17. The first version is the wild type chromosome 17, and the second version is the chromosome 17 carrying the T haplotype region. It is like a 40 megabase region in the chromosome 17. The T haplotype may always transmit the T haplotype to the about 95% of their offspring. So we ask the question, can we combine CRISPR with the T haplotype to suppress the invasive mouse populations? We insert the CRISPR system into the T haplotype region and test whether it affected its transmission. Interestingly, the T haplotype is always transmitted to the next generation by 95% in our TW2 strain. This CRISPR-Cas9 system contains the guide RNA targeting female fertility. So it shows that all the 80% of the females are infertile in the next generation. So which means this T-CRISPR is working well. Now the big question we had is, could T-CRISPR actually suppress the mouse population in the wild? So Ashigal from our modeling team ran a spatial individual-based models considering several key biological features, like how many males are mated with the one female, like how many generations are overlapping each other, how the offspring are spread throughout the population, and consider the multiple breeding cycles. This modeling system is allow us to test how the system behaves in more realistic population scenarios. Let's see how it works in the real population. The black dots shows the 200,000 mice population on an island. The red dots are the T-CRISPR, 256 T-CRISPR mice we release in the different location in that particular island. Over the time, the gene drive spread throughout the population and the entire population collapsed within about 24 years using this T-CRISPR system. This model explained that even a relatively small release of the gene drive individuals has the potential to control invasive mouse population. We published these findings in 2022, and it is the first proof of concept for a mammalian gene drive. Now we are developing island-specific version of T-CRISPR and exploring other T haplotype strategies to how we can control the invasive mouse populations. More importantly, now we have the Australian first license to generate all-in-one T-CRISPR mouse. Let's see how we're going to do that. We have selected the island. It's called Thevenard Island. It is as our study site. This island is located just off the coast of the Western Australia. Why we select this island? The mice on this island have a slightly different genetic background compared to the mainland mouse population. So Thevenard Island became an ideal location to test our genetic biocontrol strategy in real world. So we will introduce our T-CRISPR mice into the island population. And over the time, these alleles will spread throughout the island population. But it is possible that gene drive mice could escape from that island and make their way to the mainland island because the gene drive population is getting bigger and bigger in island population. The advantage of this specific version of the T-CRISPR is the gene drive will not spread in mainland mice population because their genetic background is different. Is to island population. It will not target the mainland population. For our proof of concept, we focus on a locally fixed allele that is unique to this island population. We already find that one. Now we are generating this particular version. So far, we have explored several genetic strategies to control rodent population. One of the major milestones was T-CRISPR. That is the first validated genetic biocontrol strategy for rodent population in control. We are now working on the all-in-one T-CRISPR targeting island-specific allele, which is that concept is described by the Kevin No in 2022. Now we are following that one. Also, we are conducting the enclosure trials with the CSIRO to see how T-CRISPR performs in a more natural environment. It's actually in the inside the building, but it's giving the more feeling of the natural environment, how they affect in the natural environment. Not only that, several other strategies are currently in development to control the invasive mouse population. And we had a question, what about the other vertebrates? We did modeling for the other species like rats, rabbits, feral cats, and foxes. And this is one example how Xshredder will work in these species and how they're going to affect to the control of population. When it comes to the mice, rats, and rabbits, they are smaller mammals, and their gestation period is quite small. So to eradicate those kind of population, it will take less time. When it comes to feral cats and foxes, their gestation time is quite bigger. So generally, it will take longer time to control that population. And also there will be more technical challenges to develop gene drives in the bigger, larger mammals. Not only in animals, now gene drives are available in plants as well. In 2023, these research groups published their studies, and they have done the gene drive in the plants, and it shows more than 90% transmission bias in the plants as well. So we can use this gene drive system to control weeds also. As a take-home message, when we use the traditional method like trapping, poisoning, or shooting, we need to repeat again and again to control the mouse population or invasive pest population. But the gene drive, it will spread throughout the population across the generations. The poisons can affect the other species, not only the targeted species, but the gene drive, it is a species-specific method to control the population. And also gene drive can use in places where the traditional method can't use. And I would like to thank my supervisor, Professor Paul Thomas, and my team, and all the collaborators and the funding bodies for supporting these studies. Thank you.
Thank you so much. That's awesome. Actually, the information there, as we go from smaller mosquitoes and now we're going to the bigger animals, we see that mice are really, it's awful, you know, looking at those birds being eaten alive. And I see that this is predominant in some parts of the world, especially Australia. That's why it's being done in gene drive is being tested for invasive species in there. So we'll have more questions on that. And may I ask all the participants to put your questions there. And for the, we only will entertain answers from our panelists this time. Okay, thank you so much. Now we'll have our next speaker. And so thank you so much again, Dr. Gelshan. Now I'd like to hand it over to our fourth speaker. We're really good on time. And we're excited because we will have more time for Q&A. So our next speaker is Dr. Delphine Tizzi, who will share with us about the best practices and challenges of stakeholder engagement for gene drive research. She is an independent consultant with over 15 years experience in stakeholder engagement, land rights, and global health across Africa and beyond. So let's have Dr. Tizzi now. Thank you.
Thanks a lot. Are you seeing this in the right display? Let me see. There we go.
There it is. Okay.
Yeah. Great. So I won't get into what gene drive can do and cannot do because my colleagues spoke about that very well. But one important component of gene drive research has always been the stakeholder engagement part. And I think our webinar today is part of that. It's part of making sure that the public at large is informed. And so I'm going to try to run you through a little bit of good practices and some of the challenges. But first, I wanted to really briefly discuss why to engage stakeholder in gene drive. And I won't get into all the reasons, but I thought I would point to here the fact that there can be many reasons to engage. Some, of course, are of ethical nature, about respect for the people who might be engaged in the research, who might be affected by this research, but also a question of justice and equity, making sure people have agency, being part of the decision that might affect them. You can want to engage because you want to co-develop this research, and this has been very discussed in the field of gene drive. To my knowledge, it's one of the rare fields where it's been discussed so much. So the idea that communities, stakeholders might have knowledge that they can share and that can improve the technology design, but also risk identification. You might have instrumental reasons why you want to engage, just simply because you need to get some form of acceptance or consent. You want to avoid conflict with people. You want to manage your reputation. You can have strategic reasons as well. You need stakeholders to make decisions for advocacy. You also might want to share power with stakeholders in your research process. And of course, there might be regulatory reasons. So in many places in the world, you have an obligation to have public consultation. And there we will see, because there might be different people engaging, but that could be also part of the regulatory processes. So we've established the fact that it's quite good to engage. There's quite a lot of reasons to do so. So do we know how to do it? Is there some kind of guidance? So the good and bad news is there are a lot of documents that highlight the need for a stakeholder engagement. It started all the way when WHO had its first guidance on testing genetically modified mosquitoes, but the National Academy of Science in the US, I didn't put them all here, but the one in Australia had the same. And so every publication that's been dealing with gene drive has been pointing to the need of stakeholder engagement. I want to say it's not specific to gene drive. I think it's a good practice that would apply to other area-wide vector control. So that could mean Wolbachia, that could mean aerial spraying, anything that doesn't depend on an individual decision, of course, needs let's say a more elaborate type of stakeholder engagement. And of course, things that do require individual consent still need engagement. But here it's a bit easier because you know that you're targeting the person who's making the decision for their own health, for example, case of vaccine or else. So we have clear guidance. These guidance, they all make the distinction between community, stakeholder, and publics, and establish different obligations. And I will get back to those definitions. They also all very clearly say that individual consent is not adequate for gene drive release, but actually broader than for gene drive release for any area-wide vector control. So you can't ask individual people for their decision on whether you can release these mosquitoes. It has to be a community-level authorization. That's where the good news is. The less good news is that you don't have a lot of benchmarks. So you're told these papers and guidance say we need engagement, but they don't tell you what would be a good engagement, what would be sufficient. So it's very hard to benchmark, and therefore it leaves a lot of room for debate, which is, I think, healthy, but sometimes also a lot of vulnerabilities for projects because as you don't have benchmark, it's hard to measure yourself and say that this is sufficient or not. And of course, then it's the communities that have to say that. But and we'll see that we are not always in the most good faith environment. And then. In the absence of this benchmark, that you will see there's a lot of literature from practitioners about how they have gone about doing this engagement. So this was to say, to show you the kind of sentences that you get in those guidance. They say you have community and stakeholder engagement, so they differentiate the two. It should be early, it should be phased, and this one is coming from a document specifically on mosquitoes. It should be tailored, iterative, and sustained. So all really important, but all very vague if you are a developer starting on this. I'm coming back to the fact that we have different type of stakeholders. So we talk about stakeholder engagement, but in fact, you're kind of talking about three different things: community engagement, stakeholder engagement, and public engagement. And this is coming from the National Academy of Science from the US that did this first big report called Gene Drive on the Horizon. It's already almost 10 years old, telling us how long we've been working at this. But basically, it says the communities are those that are directly impacted. So here they talk about living in a release site. We have evolved on this definition. Many of us are saying it's more about communities, is about being impacted by the activities, which can be the release, but can also be the monitoring. You have stakeholders who might have professional or personal interest in gene drive, so they might not be directly affected but have an interest. And then you have the public, the more general people that might live in that country, that might not be directly connected to it, but might have somehow a contribution to make. And so it's already three types of levels. And so for each level, you will have a different objective. They will have different interests and concern, depending, of course, how much they're affected. This means different strategies. And it could also mean it's a different type of engagement. I alluded to this earlier, that some engagement will be done by the developers, the researchers, as an ethical obligation and often legal obligation. But some of these engagements are also the government's obligation. If I take, for example, the public consultation on risk assessments as part of the regulatory process that Brenda will discuss later, this actually contains a component of public consultation and information as part of the application of the Cartagena Protocol. And so there will be an obligation to regulators. So we have to be very clear when we talk about engagement, which stakeholder we're talking about. Best practice, there are many, so I won't get into all of them, but in GeneDrive, there's been a lot of discussion about the emphasis on co-development, so making sure that stakeholders and in particular communities directly affected are part of the design of the engagement, so that they can be inputting into how they want to be consulted, how they want to make the decision. And because of this, I took back my favorite word from the regulatory part, which is a case by case. So your engagement strategy in Australia for invasive species will be different than the one in Tanzania for malaria control. It will depend on your context. It will depend also on your project's approach, whether you decide, for example, to internalize it and do all the engagement yourself, or like in the case of our colleagues in Sao Tome, to do this in partnership with the local government, for example. So you have different models. Again, they depend on your strategy and on your context. But usually all projects have been really focusing on having this locally owned and focused. So that has gone through developing glossary to discuss this in local languages, but also developing communication tools that again fit the context. And then one thing I wanted to say, and I won't we could have an entire webinar about that, but when we think stakeholder engagement, we shouldn't equate that to just acceptance or the decision making, which is, of course, a pretty instrumental part and pretty fundamental part of when people make decisions. But it's only a small component of the engagement. There are many people that will be engaged and will not be part of the decision. And even people who are part of the decision, they will have many engagement, and not all of them will be for the aim of them making a decision. I thought when starting the challenges, I would bring back my favorite slide. So if people here have seen me present before, they will know I've been using this slide for 12 years now. It's about the big challenge in GeneDrive, and in particular when you're dealing with vector-borne diseases. You basically have the decision maker in the middle. You have people who are set to benefit from this technology, and often they're people living in rural areas, being affected by malaria, for example. So they are often people living far away from the press, or not necessarily using all the social media, although we know the use of WhatsApp, for example, has increased their access, but they tend not to be the ones who are most influential. And on the other side, you have the influencers. Influencers might not all be social media influencers, but might be people who can have access to decision maker, can have a loud voice, but they might not necessarily be communities as set in the NASEM definition, as in people who are directly affected. So the whole work of most engagement projects has been to make sure that the voices of beneficiaries or affected people is heard and is at the center of the engagement, so that yes, we hear the influencers and we take their perspective into consideration, but the voices, the decision remains in beneficiaries. And so we help decision maker to see that even though influencers might be loudest, beneficiaries' voice is also very important. Many challenges, and many of them are not specific to GeneDrive. I want to mention, I could probably give the same thing talking about new kind of vaccines or a lot of new technologies. But of course, one of the first challenges is how you establish the trust, how you build that trust, which might depend on who's engaging, when you're engaging, and how this engagement is part of the broader relationship. Are you just coming one day telling them everything and then you leave, or are you building a relationship with time, taking part in communities activity and being on a longer term? So of course you would hear from what I'm saying, the best is to have a long-term relationship. I wanted to give one example. We have a paper in peer review at the moment about engagement that GeneDrive researchers have done with indigenous people. So indigenous people's representative that are part of policy forums at the global level, and how trust has been essential to doing this engagement. So a bit of advertisement here for that paper. Another challenge is the complexity. Of course, you will have heard today from the scientific jargon or the complexity. And Brenda will get to it. The regulatory process is also quite complex. Now, it is complex, and it is as complex or differently complex than a vaccine or an insecticide. But I want to say very clearly here that saying that people who are living in villages do not have the capacity to understand this and therefore making a decision is, in my view, a paternalistic position. Because I believe that, and I have experienced that, if you take the time and a really carefully thought approach, anyone can understand this. Of course, they might not understand Brian's presentation at first, but Brian wouldn't provide the same presentation if he was explaining it to a village. He would go into more details, into more time, and they would understand. So I really want to urge people not to think that this is too complex for villagers to make a decision, because there's many evidence that this has been done, and really well done. And finally, one of the challenges is balancing expectation with early engagements. You want to engage early, but you also don't want to raise too many expectations when you still have many, many years of development. I wanted to finish on a quick slide about disinformation. We are in a time of disinformation in general. We are in a time where science is under attack, and GeneDrive field is no difference. Often it's either being using, as you can see here on the right, some imagery that looks very scary, which might not even reflect where the field is at, because most GeneDrive are not for pest control in agriculture at this point. And there's a lot about making people maybe overhyping where GeneDrive is, or emphasizing specific aspects of this research that might be still at early stage, or might still be not even considered for final development. Disinformation is something really difficult to fight, especially in the age of social media and of WhatsApp, in particular in some regions of the world. It is a concerted effort that researchers and regulators, I believe, have to work together, because you don't want disinformation to impact how an innovation can be adopted. And I was in a completely different discussion this week about HIV prevention, and I was hearing the same thing. So it is not specific again to GeneDrive, but to this terrible period we are in. And I think it's a call for all of us to work together and make sure that people have the right information, and on that basis can make the decision, whether it's a yes or a no, but based on appropriate information. And here, quick takeaways to close. As I hopefully explain, I believe the engagement covers a diversity of activities, of objective, of approaches. It's very critical for Gene Drive. There's a lot of guidance existing and a large body of evidence growing from practitioners. The challenges are many, but they are not, most of the time, they are not specific to Gene Drive. And we indeed have the backlash on knowledge and expertise being more and more questioned in this time and age. Thanks a lot for your time.
Mm.
And sorry
Thank you.
for being a bit over time, I think.
No, you're good. Thank you very much, Delphine. Actually, the layout of our webinar is really very good because we started with the basics and now we're moving towards engagement. And now we're looking forward to the talk of Brinda Das on regulation and risk assessment of gene drives. The international and national frameworks. So our speaker now, Dr. Brinda Das, will provide an overview of new developments in national and international guidance and policy discussions around gene drives. Dr. Das is the Scientific Program Manager and Policy Lead for Gene Drive at FNIH Gene Convene. She is an expert in biotech policy, risk analysis, and regulatory review with prior experience at FDA and ongoing work in global gene drive policy and risk assessment. Let's have you on board, Dr. Das.
So much for having me, and I will start my presentation. Hopefully you can see the right screen.
Yes, we can.
Okay, so greetings to everybody, and today I'm going to present on the regulation of gene drive mosquitoes, particularly the frameworks for regulating them at the international level and national level. So as a background, just to highlight why governance is critical at all levels, local, national, regional, and international, is because it provides transparency to all the stakeholders. It clearly defines the requirements to get a product safely and effectively to the market and provides a level playing field for everybody, all the developers and any stakeholders interested in bringing a new product to market. This has nothing to do with gene drives. This is for any product, right? Any product that has oversight from a regulatory body. And as I mentioned, unable to move. Okay, there we go. As I mentioned, oversight regulation should be at every level. And today I'll touch on some of these levels, but just to show you that there are examples from international, regional, and national levels for the types of regulation that we might expect for gene drive, engineered gene drive products. There could be oversight from institutional ethics committees, from treaties and covenants and protocols that are international and global, regional bodies, obviously national ministries, and then at the institutional level, maybe institutional biosafety committees, right? So I'll describe some of these during my talk. Before we get into the actual regulatory part, let me just define a few terms in case people are not familiar with these. The first one is, what is a law? A law is a rule or a code of action that is binding. So there are consequences if you break the law. That's what binding means. And this law is generally enforced by some authority that has oversight or control. So it's probably a ministry and then delegated from the ministry to agencies. Beneath that is what we call regulations. In some places, you may see this as an ordinance or a decree or an order, but they are essentially all the same. And a regulation is an interpretation of the law. So this means that it has more details and it translates the law into action. So how do you actually fulfill the spirit of the law? What are the things that someone needs to do? So if we're talking about biosafety, what do people that are interested in producing a product that needs to be evaluated for biosafety need to actually provide for the oversight to the government? And finally, the lowest level is policy or guidance. And I'll have an example of that in my talk of guidance. These are typically non-binding. So they are good practices, but there are usually no no downsides if they are not followed by developers. So there are further guidance, further detailed information on how to keep in concert with the law and the regulations, right? So these are some terms that will come in handy. So in terms of actual global governance, I'll give you a couple of examples at the international level. So I've highlighted two here. One is the World Health Organization, and the other one is the UN protocols or treaties under the UN Environment Program, or UNEP. Both of these are obviously coming under the UN General Assembly. And the one I'll start with is the Convention on Biological Diversity. So I don't really have a pointer, but if you see the column that says UNEP, and under that there's some acronyms, and in parentheses and next to each acronym is a number. So those numbers identify how many countries in treaty language, countries that sign a treaty or an agreement are called parties or members. So for the first one is the Convention on Biological Diversity, which originated from the 1992 Rio Earth Summit. And its main core aim is to conserve biological diversity, its use, and fair and equitable sharing of the benefits that arise from the utilization of genetic resources, right? So this has generally grown to include other areas beyond this core concern of conservation of biological diversity, and it has some relevance to GMOs, or genetically modified organisms, which in this treaty are called living modified organisms. And engineered gene drive containing organisms are within scope because they are considered LMOs or GMOs, right? So the primary treaty here is the Convention on Biological Diversity, and then under that, there are several other supplementary agreements or treaties, and one of them, if you look at the highest level of supplementary protocols, is the Cartagena Protocol on Biosafety to the Convention on Biological Diversity. And this one is basically when a country signs onto it or becomes a signatory or a party, it is agreeing to ensure that it will regulate so that there's adequate protection for the safe transfer, handling, and use of a living modified organism or a GMO that are products of modern biotechnology, particularly for two reasons: if they may have an adverse effect on conservation, and to ensure that there's sustainable use of biodiversity, going back to the CBD, right? So this is the main supplementary protocol that has effect on GMOs, and in our case, engineered gene drive containing GMOs. And the risk assessment is described in an annex in this supplementary protocol. There are two additional other protocols that are supplementary to this Cartagena Protocol. One of them is called the Nagoya Protocol, and it deals with access to genetic resources and the benefits that accrue from them. And then there's a second supplement, which is the Nagoya Kuala Lumpur Protocol to the Cartagena Protocol. And this one deals with liability and redress related to the effects or the impacts of the LMO. So these are international treaties. And if your country, the country that you belong to, has agreed to be a party to these treaties, then obviously the terms of these treaties have to be accounted for in the national law. So the treaty requires that the countries take these on board and put them into force through national laws, right? So in terms of gene drive organisms, I will say that at the last COP-MOP for Cartagena Protocol, which was COP11 in Cali, Colombia in 2024, the parties, so majority of the countries that are signatories to Cartagena Protocol, welcomed this an additional voluntary guidance that was for risk assessment of engineered gene drive containing living modified organisms that was prepared by an ad hoc technical expert group in the time period, intercession time period of 2023-2024. So I highlight here the product now that is available to all countries to use. And this is available through the CBD website. And it just shows you the outline of the topics that are covered in this guidelines. And the material is based on Annex 3 of Cartagena Protocol, which is a high-level overview of how risk assessment should be done. Again, I will say that's just risk assessment for anything, in this particular case for LMOs, and again, further for engineered gene drive LMOs. But it also takes into account current best practices in the conduct of case-by-case risk assessment, and so it includes topics such as problem formulation and the pathways to harm approach. It's a very complete document, and I'll show you the actual decision language from CP 11.7, which is the decision from the COPMOP in Cali. And you can see here that parties welcomed with appreciation, which is pretty much a pretty high-level endorsement for the document. And then they also invite everybody to make use of this when conducting the risk assessments when the time comes for assessing engineered gene drive containing LMOs, as well as to use this for capacity building. So those who are interested in training other experts to be able to do risk assessment, this will be a good document. So this is very current information coming from the CBD side of international treaties and guidance. The other organization that I will mention really quickly, and again, this is very current to those of you who follow this, is the IUCN, or the International Union for the Conservation of Nature. And while IUCN, when it puts out a statement, it does not have the same legal weight as a treaty or a convention, but it is extremely influential in setting policy. And because it has a large membership, both government and non-government groups or organizations and agencies, it means that the decision, when it is adopted, will influence a large population of the conservation community. IUCN primarily is interested in sustainable development while also ensuring that nature is conserved. Gene drives have been a part of the larger conversation at IUCN since 2016, primarily in terms of the role within synthetic biology in conservation. So for those of you who may not have heard of IUCN, you may at least have heard of the IUCN Red List of Threatened Species. But I will say that IUCN has also played a role in many international conventions, such as the Ramsar Convention on Wetlands, CITES, or the Convention on International Trade in Endangered Species, and of course the CBD, which I just mentioned. So it actually was a big part of guiding the creation of the 3 Rio conventions, the CBD, climate change, and desertification. So it is a very influential organization. And it also has observer status with the UN General Assembly. So if you come to any UN meetings, you will find IUCN is well represented there and has a voice. And it's very influential. In terms of current events with IUCN, I will say that the IUCN has, just like COPMOPs for CBD, IUCN has Congress where members meet on a regular basis. And the most recent Congress was this October in Abu Dhabi. And there were 2 of interest to gene drives, engineered gene drives, is there were 2 motions that were up for voting at IUCN, at the Congress. The first one I'll mention, there's no particular order, is a motion 133. This is a petition that was led by NGO Save Our Seeds. This was for calling for a moratorium on genetic engineering wild species in natural ecosystems. And it was several proponents of this motion were NGOs like Polinis, Pro Natura, Nature Canada. And you can see here the actual results from the vote from both governments and non-governments that participate in IUCN, and their whole statement that the proponents of this motion were making is that nature is not a laboratory. I will say that this motion, when it was voted by all those who were present, was not approved. So this is a good thing for those who believe that safe and effective gene drive organisms can be used to help conserve nature. And the other motion that I show you here is motion 87, which is IUCN policy on synthetic biology in relation to nature conservation. And you will see that the IUCN actually agreed, and this is the first global policy on synthetic biology that the IUCN has approved. I don't have the voting numbers here, but you can see from the website that this is taken from the IUCN website, the black box, is that it was approved. And particularly what this means is that the blanket moratorium was rejected, and IUCN is instead endorsing a structured science-based framework for case-by-case assessment, stakeholder engagement, and risk analysis, which aligns, as I mentioned, with CBD and other global agreements. And this policy will cover synthetic biology in relation to nature conservation, and also other sectors like that previous speakers have said, industry, agriculture, medicine, and those areas where there may be direct or indirect impacts to nature from synthetic biology, including gene drives, right? So it's coming under a bigger umbrella of synthetic biology for nature conservation, particularly for threatened species and things that will be under threat given, the way the world is going with the global climate change and things like that. So these are very current things that are happening globally with CBD, IUCN. In the third international program that I will mention is the World Health Organization. This graphic is from the operational strategy that the Global Malaria Program put out for 2024-2030. And all I want you to take away from this is that they have 4 pillars that they want to push forward on: norms, standards, new tools and innovations, strategic information, and leadership. The thing that I want to highlight here is new tools and innovation, because this is where engineered gene drives would fit into the World Health Organization's plan for new malaria tools, right? Which are very, very necessary and needed. Don't worry about this image if it is too much for you, too many boxes. All I want to say is that engineered gene drive mosquitoes do not have a clear existing WHO policy recommendation at this point, but that doesn't matter, because there is actually a process that the WHO has where a policy recommendation can be reached through various advisory groups. So if you look at the left side, the column on the left side, there's an advisory group called the Vector Control Advisory Group. These are independent experts that come and support the WHO by looking at the data, stuff that people like Brian are doing in the lab, producing data. They can submit it to the VCAG for their consideration. VCAG makes a determination that it has a public health value. This is confirmed from the data. And then there are various other advisory groups that are internal to the WHO that now work with a guidance development group to actually develop a policy recommendation, which is made available to the public, to developers, to everybody else. Then on the right side, you can see that once you have a policy, there is a group within WHO called PQ, and that stands for pre-qualification. And they will then look at the actual product, the quality, the safety, the efficacy from start to finish, including manufacturing. They confirm all of that, and then this product can be listed and then made available for procurement by those who are interested in using that product distribution, right? Some of this has already been covered. The WHO has also put out a lot of guidance documents. These are all available publicly on the WHO website for anybody who wants to look at them. There's guidance framework for testing of genetically modified mosquitoes, how to design vector control trials, how best practices for, the testing of safety and efficacy of gene-derived mosquitoes, particularly in our case for malaria control. And they show a phased pathway that starts with containment, which is where Brian's group is now, which is laboratory only studies. And then as you prove safety, you will move increasingly to less containment and towards field testing, field release, and greater environmental and human exposure. Moving down one level from international, global to more regional consideration, our interests are right now with malaria control using these new tools. And there are groups that are working across Africa. One of them is the African Union Development Agency, New Partnerships for Africa's Development, otherwise called AUDA, NEPAD, or AUDA NEPAD. They have an IVM platform. They have put out several guidance documents to help the African countries, particularly in the western part, so the West African IVM platform. And they are working with that group of countries to come up with a regional focus so that when a product gets to the point of being released for testing in a field setting, there will be regional acceptance because these mosquitoes are expected to spread and disperse. So if all the neighboring countries are prepared, then it will be easier to do the next step, which is a field release and field testing, right? And finally, I'll just briefly tell you that from most countries' perspective, especially parties to the Cartagena Protocol, typically the national system is a stacked system like this, where a developer will produce a dossier, will produce a desire to work with an institution. So they will work with the institution to put together a dossier. The group within the institution that will be most involved with this, other than the researchers, of course, is the Institutional Biosafety Committee. This is the group of people who will then package all that information in a way that is satisfactory to the competent authority of that country. Typically, it's called a National Biosafety or National Competent Authority. This group will then assess all the information, and then with the help of its governing ministry, which could be environment or health or agriculture or combination or science and technology, they will have all the ministerial input and a decision will be made in keeping, if they are a party to any international treaty, in keeping with all of those treaties and the national laws. So that is how the decision making goes up. But then once a decision is made, that decision is shared all the way back down through these organizations, right? And I just want to leave you with this slide that there are a lot of parties that have oversight. There are a lot of people and groups that have roles. Everyone that you've heard from today has covered some aspect of all these different players. So I'll stop here. Thank you.
Thank you so much, Dr. Das. Actually, it's very interesting that we have new information about the decisions in the IUCN, and we're happy to note that everything's well, and we are going to go for business as usual. I mean, we will continue on with all of our research and engagements. Okay, thank you so much. Actually, we are almost running out of time, so I can only entertain a few questions, and we have a few questions on the board right now for the Q&A in the Q&A chat box. So can we have all of our speakers, can you turn on your cameras so that we can have this discussion all together. I'm only able to raise 3 questions at this point. So let's have first Delphine, Brian, and yeah, okay, we're missing 2 more people. Can you open your cameras, please? So the first question that came up was answered by some of the panelists and outside the panel, but the first question that we can start could be from where's Jackson? Jackson, are you there? Okay, the first question will be for Jackson and include Jackson in the spotlight. Could you elaborate on the timeline for moving from laboratory research to potential field trials and what key milestones need to be achieved? I think this question is also for Dr. Gelshan and Dr. Brian. So an estimate of how long would it take to move from laboratory to potential field trials, and what key milestones need to be achieved. So let's have Dr. Jackson first.
Well, in the laboratory, at least, we have many different ways to assess a gene drive and measure its performance. In order for a gene drive to be considered for field trials, it will need to have good performance in individual crosses between insects, and then in cage studies that are designed to simulate a gene drive's performance in the wild over multiple generations. And then if it's good in a cage study, we'll use computational modeling and our best possible predictions and data to see if this gene drive would work in a wild situation. And if all of this checks out, then we're ready to file for a plan and approval to actually release these gene drives. This can take an extremely variable amount of time based on the scientific research, since different species and different gene drives will have completely different problems to overcome. CRISPR gene drive research has been going on for the past 10 years, and so far there are no release candidates. But some release candidates are getting very close in Anopheles malaria mosquitoes, and it's thought that once the laboratory research is finished, it will be perhaps within a few years that the actual releases can take place.
Okay, yes. Okay, can we hear from Dr. Gelshan and Dr. Brian? One at a time. Sorry, Dr. Brian first.
I mean, I'll let Dr. Gelshan go ahead because my answer will be pretty much what Jackson mentioned. He pretty much covered what the gene drives for some species and definitely for mosquito vectors. So, well, go ahead, Dr. Gelshan.
Yeah. Like Jackson explained, it will be different with the different species and different things. For the mice, it will take quite a longer time to develop the system in the lab, not like the insects, because their generation time is much longer compared to the insects. And also, it is, as I explained, it is not easy to copy the gene drive, which is establishing the insects into the mammals. Their system is the in vivo system is completely different. We try to copy the same as the insects. In the homing gene drive, it didn't work. So we had to try the different timing of the Cas9. And so it will take a lot of time to develop in the laboratory scale. So then only we can try to the real world. So now we actually trying the enclosure trials in the CSIRO. It's like pretty similar to the wild, but it's not exactly same for the, especially the T-CRISPR, because that system is working in the lab. So we will get more data from the enclosure trials. So it will take longer time. Yeah.
Yeah, thank you so much for your insights. Now, there's a question from Rudiger Stegemann. This is for Brinda. Brinda or Brian, what about governance frameworks? No, this is for Brinda. What about governance frameworks given by UNDROP, UNDRIP, and other HR related instruments. I think this is for the laws and for how it is going to be governed by different countries.
So already Delphine provided an answer in the chat, so I won't repeat that part, but I would say that from my perspective, the voice of the IPLCs will not be neglected. I mean, I would not just restrict to IPLCs. Stakeholders who are in the use areas, testing areas, release areas, they will be included by the governments as well, by their own government, local and national, as well as the stakeholder engagement, the people who are doing stakeholder engagement, to make sure that their concerns are met. And if their concerns are not met, then the work will not proceed. I don't see how the work would continue. And also, the country laws should include all of these considerations that Rudiger has mentioned there. If the country laws have taken these on board, then the developers will have to do what the law says. So if there's a specific call out for IPLCs, or there's some specific tribe that has valued species or whatever, when they do the problem formulation, part of problem formulation includes reaching out to the stakeholders, and they would definitely be a stakeholder. So I don't see why their voice would not be heard.
Okay, yeah, that's correct. And Delphine, may you repeat what you have written in the Q&A box for our participants to know?
Yes, sure. So the different frameworks is mentioning and other human rights, and so people understand the acronyms, human rights instruments. Basically, a lot of the existing governance frameworks are making direct reference to them. So the decision of the CBD on gene drive release has a specific paragraph about the rights for free, prior, informed consent. The WHO also refers to rights-based approaches, and of course, all stakeholders' rights need to be taken into account, but there are specific safeguards for indigenous people. So this is integrated in a lot of the frameworks. And then when it comes to projects and how they're looking at the governance, as Brenda said, if you are in a country that has presence of indigenous people, then it's likely to be reflected in the national laws. So you will come and see what are the specific provisions for that. And of course, this would trigger a lot of different aspects. So it would trigger things if you're doing an impact assessment. It would trigger specific approach to stakeholder engagement, because indigenous people have specific protections in this. So you would still do the same engagement, but with specific rights. As I said earlier, you adapt to context. So it's a very important question that Rudiger is pointing, and it's very context specific. Some projects might not have indigenous population in the area where they're currently working on.
Yeah, that's right. Okay, now let's move back to our other speakers. This is now for Brian, I think. The question is from Bob Phelps. The World Mosquito Program's Wolbachia program has reduced dengue in Northern Australia, very good, to zero in the past decade. Reduced to zero. It appears less harmful to mosquito-dependent organisms in ecosystems such as bats, fish, birds, and dragonflies, as it does not crush mosquito populations. Instead, it disrupts the mosquito's ability to vector diseases. So are gene drives still needed for mosquito control or eradication at this point with these alternative measures?
Okay, so he's talking about Wolbachia for the control of dengue. So quickly to start with this answer is there are over 3,000 different species of mosquitoes. The mosquito that transmits dengue is not chikungunya, Zika, yellow fever, and these other arbovirus is not the same mosquito that transmits malaria. Aedes mosquitoes are the ones that transmit dengue, and malaria is transmitted by mosquitoes that follow under the genus Anopheles. So currently there isn't any, you cannot Wolbachia in Anopheles for the control of malaria. There hasn't been any release of that, or even if you can even implant or put Wolbachia in Anopheles for the control of malaria. So what I can say with regards to genetic-based control strategies for malaria, the Wolbachia endosymbionts might not necessarily work as it works with Aedes mosquitoes in the control of dengue, chikungunya, and other arboviruses. And also Jackson and myself, when we were explaining about gene drives, we talked about two different strategies. So there's the population suppression and population modification. Even with the suppression, no one is talking about eliminating a species, but we're talking about reducing a species to such a low number that transmission is severely hindered or stopped. So none of the two strategies are talking about elimination of a species rather than reduction to level that transmission is prevented. Thank you.
Thank you so much. And will that be the same as well for bigger pests, especially for invasive species? It's also for suppression, not the eradication, right?
Yes, yeah.
Yeah, thank you, Gulshan. Actually, we are running out of time, really running out of time. So I will give you. Yeah,
Just
no, I mean, okay, go ahead.
So you suppress the species. We want to eradicate the disease. We want to suppress mosquitoes to a level, but we want to eradicate malaria. Or in the case of bigger rodents, it's the same thing, I would assume.
Okay. Yes. Thank you so much. So once again, may we give a round of applause to our speakers. We're running out of time. And to synthesize, I think we have already seen what's happening on Gene Drive. We have started with the basics, with the sciences, and then we went to the engagement and also how we are dealing with Gene Drive and synthetic biology. And now it is accepted and we can continue on. There's no barriers anymore. So we are looking at a way of reducing our diseases through this Gene Drive, suppressing the disease, and also for eradicating some of the invasive species that hinder the production of food and the production of agriculture, and that will help the humankind to thrive in our world. So let me ask everybody to give a round of applause, virtual applause, to our speakers. And can we have now the last slide, which is the QR code for the evaluation form. So we are now going to ask all the participants to evaluate our webinar. And as soon as you respond to this evaluation form, there will be certificates going to your inbox in your email. So use the email address that you would want your certificates to go into. So I would like to thank everyone in this webinar, especially the Gene Drive Network. We have our help from Eva and from Zaira for the questions. And of course, all our speakers are here. You can contact them. If you have any other questions, put them in the Q&A box. We're still going live after 15 minutes, waiting for your questions. And for those who have still not satisfied with all of these questions to be answered, we will get them to the questions to our speakers tonight. So thank you. And with that, I'd like to say goodbye, and let's have another Gene Drive webinar in the next year or so. As we move forward, we are really looking at the release of these materials, of the Gene Drive materials, not only for health, but also for food and agriculture. Thank you so much, and have a good evening, good day, everyone. Thank you. Thank you so much.
Thanks, Brian. You're free to go.
Thank you,
Oh,
Brian.
There's a question I was trying to answer. Or I should just
Oh,
leave
okay.
it.
Yes, please go ahead. No problem.
Okay.
We're going to have this on file, the questions as well and the answers.
I send to everyone or privately to
Hmm?
I send it privately to the person who asked. Or I can send to everyone. But they're out, so they might not see it.
Just put it there to everyone because we'll have this on file. This is recorded. Yeah.
All right.
Thank you.
Thank you. Bye-bye. Have a great day, guys.
Thanks,
Bye.
Brian.
Thank you, Brian.
Bye.
Okay. Are they all out? We reached up to how many? I recorded around 111.
Yeah.